We just the three of
Today
us.
I want to expand my original title a little bit and talk about the network-based methods I and my colleagues at the Lage lab have been developing to interpret large-scale genomic data sets. So the first part I want to talk about EMAP, or EMAP-IM for short, which is the high-quality protein-protein human interaction network, just published in Nature Methods. And then we'll introduce some of the ongoing work on GeNets, which provides user-friendly tools to help you tailor network methods for everyone's specific use, to make it easy for you to use some of the methods I'll talk about in the first paper. So to start with, I want to remind everyone of the basic concepts of biological networks. So we're now equipped with very advanced techniques to generate a lot of data, diverse types of functional genomics data, from gene co-expression correlations all the way to more in silico-based co-mentioning in text kind of data sources. And we can summarize all of these data in the form of networks, where the genes are the vertices in the network, and the edges between them represent any kind of functional associations between the genes. And this kind of representation of the data, a very complicated data set, can lead to the discovery of biological insights that would otherwise be missed. And so in today's talk, we'll focus on protein-protein interaction networks, second source here. The reason why I want to focus on protein-protein interaction, or PPI, today is because the physical interaction networks data can complement, for example, the epigenetics or RNA sequencing data you get from your studies, and provide additional dimension, important biological insights for your particular studies. And here we'll talk about two examples where you can use the PPI resource to help your understanding. The first example is about congenital diaphragmatic hernia, or CDH. CDH is a birth defect. Essentially, there's a hole in the diaphragm which allows the organs from the abdomen to move all the way into the chest. Although the outlook for babies born with CDH has been increasingly positive because of the improvement in the surgical techniques, we still don't fully understand the molecular causes of this disease. So in this study, the authors identified likely pathogenic variants from exome sequencing of about 200 trails, and they found and prioritized the genes with very rare variants using the PPI. So the green ones are the ones that they were able to find using the PPI methods. Further, they identified several of the very coherent subnetworks of interacting proteins predicted to be important in CDH. For example, the muscle development pathways, cell migration and signaling pathways, and a lot of the transcription factors with specific roles in cardiac development and blood vessel formation. So this data have been able to help researchers dissect the very heterogeneous etiology of CDH, this disease. The second case study I want to talk about is autism spectrum disorder, or ASD, which affects about 1 in every 68 children in the US. And many large-scale studies have been dedicated to identifying the risk variants for ASD. And this study integrated data from several large consortia, about thousands of patients and controls. They were able to zone in to found 65 autism risk genes shown here. What the authors were able to do is they embedded the 65 autism genes in protein-protein interaction network, and they found 2 subnetworks that are enriched for genes. Here on the left are enriched for genes in sort of chromatin regulators, and here on the right, the proteins are enriched for genes in sort of synaptic proteins. And this kind of study have already helped direct some of the follow-up experiments and studies in autism. And I will come back to talk about the 65 genes in a future slide. So here, I just want to emphasize that, as these 2 examples have shown, the PPI-based methods have been really cost-effective in scalable way to help you identify cellular networks and enrich the genetic risks. And you can design your further studies based on these results. But all such analysis require reliable PPI network. So the key question, therefore, is which one among there are many existing resources out there, which one should we use? So here we built a comprehensive and high-quality human PPI, InWeb, InBioMap, or InWeb IM, which I will show outperformed other widely used resources by several important metrics. The network is called InWeb because we borrowed ideas from earlier versions of InWeb. The network, we started with 25 million articles in PubMed, which are cataloged in 8 different PPI databases: NetPath, Bind, BioGrid, MatrixDB, Reactome, Intact, DIP, and Wikipathways. We also extracted raw data from 57 organisms, used majority voting of 8 of the authority transfer databases, transfer data from other organisms. Essentially, if at least 4 of the 8 databases agree on this particular transfer, we put the data from other organism into human. And the rationale here is that such a stringent authority transfer scheme, we can increase both the coverage and the accuracy of the resulting data. Because high quality data from other organisms can effectively increase the signal-to-noise ratio in the human data. And we scored each of the interaction, which is the scores are based on the quality and the quantity of literature support, and transform the scores into a confidence score from 0 to 1 with a probabilistic meaning. For example, if you have a score of 0.5 in our network, that means at least there's at least 50% probability that this interaction is true positive. Believe this kind of scoring scheme will help users sort of filter their interactions and integrate this network for Bayesian inferences. So at the end of the day, we have a network with well over 500,000 interactions and spanning 17.5K human proteins, which is about 90% of the reviewed part of the human problem. And on top of that, we provide complete transparency of the data source. And by that, I meant if you compare InWeb IM with 5 other widely used PPI resources, you will find that we're the only one that consistently provides all the detailed information for each of the interaction. We score to the publication that supports each of the interaction, so which one of the source database that interaction is extracted from, as well as which organism that data is transferred from. Here we show that InWeb IM is the largest network in terms of the number of interactions, as well as the number of publications supporting these interactions. And most notably, we draw data from 34.1% more publications compared to the second largest network, I2D, PINA, in terms of the coverage. And in terms of authority transfer, 68% of the data comes from either human or mice, and about 95% of the data comes from human, mouse, rat, cow, nematode, fly, or yeast. Also, a large proportion of interactions, more than 50% of InWeb interactions, are unique to InWeb, which could explain some of the uniquely enabling properties, which I will talk about later. So we have seen that InWeb is a very comprehensive resource, and how do we know it has high quality interactions? Well, first we obtained data from independent proteomics SciLAC experiment from 58 human immunoprecipitations, which this is not included in any of the networks, in any of the 6 networks that we looked at. And we chose the 3 networks, InWeb, Menta, and IREF, which has scores, which is correlated the heavy to light ratio, isotope ratio from the experiment to the confidence scores of the interactions in each of the 3 networks. So here the heavy to light isotope ratio is a rough measure of the confidence of interactions between the proteins. And here we can find that InWeb and IREF actually have a very high correlation between their confidence scores and the experiment independent data set. And next, we asked whether InWeb had better biological signals compared to these other 5 networks, which is, I think, really the need of this paper. And we did 3 tests here. And the first test we did was we gathered data from 853 pathways very stringently defined from the molecular signature database, or MSigDB. And for each of the pathways, we hold out 60%, 30% of the pathway members. And we asked each of the network based on topological features of the 70% genes in the 70% of the pathway members, the topological features of the 70% of the pathway members, can they recover the 30% left out genes? We found that InWeb has an area under the receiver operating characteristic curve of 0.95, which is the highest among the 6 networks, which sort of convinced us that InWeb is the best network at learning pathway relationships. And this particular algorithm, which we call QUAC, we'll come back to talk about this when we talk about the GeNets portal, the later part of this talk. The second test we did was to try to interpret large-scale genomic data sets. So an earlier study had sequenced about almost 5,000 cancer genomes from 21 tumor types that identified 219 significantly mutated cancer genes. And so here the idea is really we want to see if, based on the interaction partners of each of the genes, can we also infer the cancer driver gene status of the gene itself. So this algorithm is called network mutation burden, where we essentially aggregated the mutation frequencies of the interaction partners of each of the genes and try to see if we can, so given the mutation frequencies, the interaction partners of the genes, we score each of the genes and we correlate that with the cancer driver genes detected from the sequencing study. And we have shown that InWeb is the best at distinguishing the 219 cancer driver genes simply because they have more significantly mutated interaction partners in the InWeb network. And we'll later use this network mutation burden or NMB approach to study autism genes. And this particular test shows that InWeb has potential to uncover genes with medium mutation frequencies because you can aggregate the mutation frequencies of the interaction partners, which effectively increase the power of gene detection in these kind of sequencing studies. The last test we did was to assess how well each of the network agrees with the data from the independent proteomic experiment, which I talked about earlier. This time we tried to use the topological features of each of the network to predict the 15,000, roughly 15,000 interactions that we discovered from the 58 pull-down experiments. We found once again that InWeb showed the best overall concordance with this independent experiment. And because InWeb has consistently outperformed the existing networks in these 3 independent set tests, we therefore were pretty convinced that it had the optimal biological signal and should be a valuable tool for network-based analysis for genomic data sets. But to further explore its potential for InWeb to inform biological studies, we integrated it with tissue-specific expression quantitative trait loci or EQTL data from the GTEC consortium we derived 27 protein-protein interaction network where the corresponding genes have a tissue-specific EQTL regulation. So we essentially derived roughly 27 tissue-specific networks. And here we show the color bars show the relative numbers of the interactions in proteins in each of the 27 tissue-specific networks compared to the second largest or the medium of all of the networks. As you can see, all of the color bars are above 1, which means InWeb has more data in each of the 27 tissue types compared to the rest of the 5 networks. And one of the most notable examples here is the brain, in which InWeb has more than twice as many data compared to the second largest network. So we hypothesized that InWeb will be better at modeling neurodevelopmental disorders like autism. So we took the same 65 genes identified in the Sanders paper I talked about earlier, and for each of the 6 protein-protein interaction networks, we rerun the NMB approach, which I remind you is an algorithm that uses the aggregated mutation frequencies of the interaction partners to try to infer the mutation frequencies of the genes itself. So we just try to see to what extent the 65 genes, which we know are sort of related, implicated in autism can be distinguished from the A and B scores in each of the networks. And here, as you can see, also the blue diamonds here are the 65 genes, the AUCs, the 65 real autism genes. And this distribution shows the 120 random gene sets, degree matched to the 65 autism genes. And InWeb is the only gene network that can distinguish the real autism genes with the random controls, which sort of confirmed the potential for InWeb to study psychiatric disorders. And I'll come back to this point later. Last but not least, we're committed to continuously maintaining and updating InWeb. This resource is already freely available for all academic users. You can find the link. You can also visualize some of the network using this Intomics link, and also using the GeNets web portal, which I will talk about next. So I'll switch gears now to talk about our ongoing work in GeNets, which provides user-friendly web interface where everyone can perform this kind of network analysis based on their specific project needs. And this is the welcome page of the web app, and here's the link here. As you can see, we really want to make it easy for everybody to analyze, visualize, and manage your projects using biological networks. And I'll talk about 3 features of the web app, GeNets, and then we'll provide a case study to show you some of the typical workflows of how you can use this web portal. So the first feature of GeNets is that it enables you to compare and choose the best network for your particular study. Although here we focus purely on PPIs, GeNets have preloaded 5 other types of networks that you can use. And it is very easy for you to upload your own favorite network. You can then train your network-specific quack model, which can assess the ability for each of the network to learn pathway relationships, given the 853 canonical pathways in MSigDB. And we also provide you with a pathway-specific score, so that you can choose specific networks based on the scores on a particular set of relevant pathways. So for example, here is the compare models page in GeNets. The heat map here shows on the rows here are each of the networks, and the columns here are each of the pathways. And for example, if you're studying psychiatric disorders, you might be interested in some of the neurodevelopmental pathways. You can choose a subset of neurodevelopmental pathways and focus on particular AUC scores of each of the networks. And if your network has a consistently higher score in the relevant pathways of your interest, then you can definitely just go ahead and choose that network for your study. This table summarizes for the PPI network. You can click on here, each of the network, that the table will summarize the scores with each of the pathways ranked. The second feature of GeNets is that it will enable you to study your gene set in a very integrated manner. So given your input gene list, GeNets can screen additional genes that are likely to be involved in this particular gene set input in the same pathway. And also you can easily annotate resulting genes using orthogonal genetic data sets. And lastly, GeNets makes it easier for you to store and share your results. Here we take security very seriously. Your private analysis will only be visible to you, and you can share results, and that results will only be visible to people with the links. But we'd also provide a pre-trained analysis using the GWAS data sets as well as the cancer sequence data, which is accessible to everyone. You can start with them to play with it. So now I will demonstrate a sort of a typical workflow that exploits some of the unique GeNets features. We use this platform to study the 65 genes implicated in autism spectrum disorders, which we talked about earlier. So first, to choose the best network to study the 65 autism genes, we tested which of the network has the best signal across the pathways that are involved in neurodevelopmental processes from MSigDB. So here, on the x-axis is the list of pathways that we believe are very important in neurodevelopmental processes. Here is different networks preloaded in GeNets. As you can see, EWAP performs consistently better at learning these pathway structures compared to the rest of the 4 pathway networks. Here's the distribution, sort of the same idea, just turns heat map into distribution bar plot. And again, the EWAP outperforms the other 4 types of networks. So we decided to go ahead and use EWAP for this.
What does learning actually mean in that case?
This learning means if you leave out 30% of the pathway members, can you recover the 30% based on the topological features in this network on the 70% of the pathway members? And here we show the quick visualization feature of GeNets, where you just show the direct protein-protein interactions from EWAP of the 65 genes in the Sanders paper. So upon clicking into the edges, you can see this window, and you can go directly to the publications supporting this interaction. And if you click on each of the genes, you can also get additional information about that gene. And here, notably, we also integrated the exact score. Here, the exact score, which reflects the intolerance of that gene to missense loss of function mutations, or essentially how well that gene is conserved. So we think that these kind of external information will help you just get a first glimpse of the data set, and you can explore further this information if you want. And we then ran the EWAP specific quack model on the 65 seed genes, and we identified 31 genes, candidates that are also likely to be involved in autism, based on the topological similarities between with the 65 genes. So here the blue ones are the seed genes, and the green ones are the candidates that we developed, that we uncovered using the model. And some of the candidates, as you can see, connected these seed genes, which just gives you a rough idea how sort of what the algorithm is working to try to find these candidates. And next, we use the GeNets annotation feature, which we call the easy encode feature, to indicate the genes that are uncovered from the genetic studies. So the red genes are implicated in the schizophrenia GWAS studies, and the blue ones here are implicated in the neurodevelopmental phenotypes, neurodevelopmental delay. These 2 data types are sort of independent genetic data sets that are used here to enrich our network analysis, proteomic analysis. And as you can see here, CTCF, NAGA, as well as SGSM2 overlap are the overlaps with the candidates. So these 3 are among the 31 candidates that we discovered using the quack algorithm. And here the hypergeometric p-value, the overlap is 0.04, which just supported the fact that quack has the ability to predict proteins that are relevant to neuropsychiatric disorders based on the seed genes from autism studies. And to further explore the EWAP quack autism candidates, we applied the easy encodes feature. And here we annotated the genes that are under brain-specific EQTL regulation. So the larger nodes here have a brain-specific EQTL compared to the smaller ones. And once again, the independent genetic data set and the EQTL data converge on this NAGA gene, which gives us confidence that it might be a very interesting autism gene candidate. And in fact, we're very pleasantly surprised to find that NAGA provides instructions for making lysosomal enzyme responsible for breaking down glycoproteins and glycolipids. And mutations in NAGA gene has been implicated in Schindler's disease, which has overlapping symptoms with autism, such as impaired communication and socialization skills. So we're very excited to further pursue this particular candidate. Although this is just a very preliminary study, we believe as more, I just want to give you a flavor of how you can use this web portal. And we believe that as more network data and genetic results become available, GeNets will become an increasingly useful tool to the community, and just provide in general a very standardized tool for you to perform your network-based analysis of large-scale genomic data sets. And with that, I want to thank Casper Lage and my wonderful lab mates at the Lage Lab and GH Broad. Here, David, Tim, and Lerat at the Broad KDocs team has helped with developing the GeNets web portal. Intomics and its CBS and DTU people at the Danish team has been really helpful at setting up the pipelines for building up the eBiomap resource and helping to maintaining it. And I'm happy to take any questions, everybody. And thank you so much for your attention.